All right, so sometimes these regulatory networks and transcriptional responses can be huge or elaborate. In the case of catabolite repression, a limitation in carbon, specifically, not the absence of glucose, can affect all of these genes. Now, catabolite repression is actually when you have a lot of glucose. So this is kind of, the image shown here is kind of the opposite in terms of at least the arrows, but the network is the same. A low amount of glucose leads to, upregulation here, and these genes are repressed, are downregulated or upregulated. And there's a lot of them. And there's a lot of these kind of global regulators, because if you're a cell and you've got, just like when we analyze these pathways, we know for a while that there are all these stretches of pathways that are linear or that perform related functions that we want to group. And so it makes sense to have some kind of controller, control valve, just affect what's going on through the whole group rather than just individually. And so you can see that here for a number of different regulators, and there are a lot more than just this. It can be useful to understand what gene regulatory networks look like visually, and to really be familiar with this sort of representation, where typically circles or nodes are your different transcription factors, or they're your genes, and they can cause inhibition. And this is the same kind of notation that we see for things inhibiting promoters that I might have brought up earlier in the course. And in this case, the arrows are activation. And you can see that here. So I want to get into metabolism now that we've covered some basics here. I mean, the lac operon is also metabolism, but it's more carbon source oriented and kind of classical and interesting for its complex logic. It's sort of two-factor, regulation. But let's talk about some real biosynthesis pathways that may be used a lot for metabolic engineering. And I come to amino acids, which is also one of the topics in your set of group projects. So amino acids, really important metabolites, especially made via fermentation, chiral, and building blocks, not just to proteins, but all kinds of other molecules. From a cell's perspective, a lot of those same reasons that I mentioned why we might be interested in them, cells are really interested in, especially when you've got proteins that are being sometimes, a cell experiences some kind of stress, all of a sudden it's turning on a lot of genes and it needs to make a lot of proteins. It has to be able to very delicately sense how much of my amino acids do I have around, and do I have the right balance to be able to make the proteins I need. When you have that kind of a situation, which appears all the time in nature, hey, I've got this critical metabolite whose concentration that I need to keep in homeostasis. You find that those pathways have negative feedback. And in fact, in a lot of human diseases, when your negative feedback loop breaks, that's what can cause a disease, because then you end up making a toxic amount of some molecule. So negative feedback is where the product of the pathway, or it can be a downstream metabolite, exerts negative regulation on an earlier pathway step. And here I mentioned the homeostasis part. So if we look at aromatic amino acid biosynthesis, we've got E4P from pentose phosphate pathway and PEP from glycolysis coming together, condensing to form this DAHP, which is the first committed step to aromatic amino acids. And what I mean by that is once these two come together, there's no going back, and there's really no other kinds of products that this molecule is used to biosynthesize. So it's not yet aromatic. It's got this heterocycle here. And after 3 steps, it's still not. Here we're at shikimate. This is often called the shikimate pathway. Later on, we get to chorismate, and you guys saw this, at least textually, maybe not visually in terms of structures, in your last homework. But I recall probably including a couple of these structures for you in the first homework to draw. Hopefully, it all feels like things are coming together. So then at chorismate, you have this divergence towards tryptophan or tyrosine or phenylalanine. Okay, so in this pathway, this is what appears to be a somewhat blurry drawing from a paper that I probably should have cited here, that shows different kinds of regulation occurring at stages of this pathway. And so it's just to reorient you, here's PEPs, E4P, you've got your DHP, chorismate, tryptophan, phenylalanine with the typo, tyrosine. And what you predominantly see is one kind of regulation over and over again. There is no step shown here subject to positive regulation. This is all negative regulation. And interestingly, it's not just your final metabolites going back to the first committed step that is also happening, but you've got these intermediate layers of negative regulation, really trying to make sure that the cell doesn't, that you have multiple layers of control. Maybe it's in case this fails, but it seems more complicated than that. It may have a lot to do with these branch points, because say you've got a really limited amount of tryptophan, and so your cell needs more of that. You don't want to have these final products all just affect DHP because, rather it's synthetase, that's what the S is for, because then you have this valve still turned off, essentially. So what you might have instead is different kinds of regulation to where maybe you can allow some flux through here and still hold off the conversion of chorismate to propionate. And that way you allow things to deal with their different branch points. And you see that same principle applying here from the propionate precursor down either of these routes. So I want to spend a little bit more time talking about this example because I think it's really neat and it's useful from an engineering perspective. In my lab, we've had to think about it. So feedback inhibition, the first committed step, whether it's aromatic amino acid biosynthesis or other pathways, is usually the one regulated, but you could have branch points as well. If we look at the DHP synthase, actually E. coli makes 3 different isozymes of that enzyme. And each one is sensitive to one of the 3 aromatic amino acids. They all seem to function by mostly competitive, in one case, competitive and non-competitive inhibition. And so that's actually happening at the enzyme level. That's an example of post-translational regulation, where you don't have to actually change how much of the enzyme you have around. If you think about it, that's kind of nice. You can just, your cell, you can just make the enzyme, and then it can allow the amino acid concentrations to just kind of naturally, if there's too much of it, inhibit or not. But you've also got this other layer going on. You've got 2 different regulators, and these are images from EcoPsych, that are repressing the biosynthesis, the formation of these isozymes. So ARO-G and ARO-H are respectively the isozymes that would work for either phenylalanine or tryptophan. Or rather, I should say, they all perform the same function. They all make DHP, but ARO-G and ARO-H are respectively the ones that are sensitive to either phenylalanine or tryptophan. And so they're also, their transcription is also regulated by this TYR-R or TRPR respectively.